third organic molecule or class of organic molecules I want to go over with you are proteins, and this would be learning outcome D15. Proteins have a wide variety of functions within the body, probably the most diverse group of functions. They are structural. In other words, they act as building blocks, as in muscle cells. They are the majority of muscle cells are proteins. They play a role in communication. They can act as various hormones and neurotransmitters, so allow communication to occur with the body. They can play a role in transport, transporting substances into and out of cells and through the body. They can facilitate chemical reactions in the form of enzymes, and they play a role in recognition and protection of the body. And we'll address some of these as we go through the different chapters, so I won't get into all of them. I will actually address enzymes in this video a little bit. Now, in order to understand proteins, we need to understand the basic structure of proteins. And proteins' basic building blocks are amino acids. And so amino refers to the fact that it has a nitrogen-containing group on it. Remember that functional group? And acid, because it actually has a carboxyl group associated with it, which is a C double bond with an oxygen. And why they don't have one here, not quite sure. So when we look at amino acid, what we find is we find what we call an alpha carbon. And bonded to that alpha carbon is the carboxyl group, is the amino group, is a hydrogen, and then we also have what we call the R group or the radical group. Now there are approximately 20 amino acids that are important for the proteins in the human body. 12 of these are naturally synthesized, 8 are what we call essential amino acids, and meaning that we have to get them from our diet or from other sources. In other words, our body will not make them. Now, the difference between these 20 amino acids is going to be this radical group. All right, so I do want you to memorize and understand the basic structure of an amino acid in general, but not all 20 different amino acids. So this radical group could be another hydrogen. It could be what we call a benzene ring, which is a 6-carbon, that's not really a pentagon, a hexagon, but a 6-carbon molecule. There's a variety of different things that radical group could be, and that's what differentiates the 20 different amino acids from one another. Proteins are kind of unique in that some proteins are hydrophobic, some are hydrophilic, and some parts of proteins can be hydrophobic and some parts can be hydrophilic. Proteins are also unique in that they are subject to what we call denaturation. In other words, changes in pH or temperature can actually cause them to break down. And so proteins have to have, they're found in a very, very specific optimal window of pH and temperature. Otherwise, they don't function like they should. And when a protein denatures, it changes its shape. Remember way back at the beginning in the first video I talked about form and function? Well, proteins are a perfect example of form and function. The structure of a protein really determines what it does. And if something happens to the pH that protein is associated with, or the temperature where that protein is found, that can actually break some of the bonds within that protein and ultimately then change the shape of the protein, and therefore that protein is then going to not function as it should. Alright, let's look at the structure of proteins. There are 4 different levels of structure within a protein. They are called primary, secondary, tertiary, and quaternary. Primary structure of a protein is pretty simple. It's just the sequence of the amino acids. In other words, the order of those amino acids. Amino acid A, B, C, A, B, C, A, B, C, whatever the case is. Okay? That's simply the primary structure of the protein. Secondary structure refers to how those sequences of amino acids, based on their order, how they actually cause, they may trigger either a bending or a folding of that chain. All right, so this refers to either the bending or the twisting. I like twisting better than folding. Bending or twisting of the amino acid chain. And what we find is that there's two different secondary structures. There's what we call an alpha helix, which is like a spiral. It's like half of a spiral staircase. And we also have what we call a beta sheet or beta pleated sheet. And that's like folding a piece of paper and making it and unfolding it then so it looks like a set of steps. So here's our alpha helix, here's our beta sheet. And these form as a result of the chemical and physical properties of the different amino acids and the way that they are arranged. And as a result of that, the individual amino acids can actually interact with each other by forming hydrogen bonds with one another. And so what we find in a beta sheet is that we have hydrogen bonds forming, this would be your beta sheet here, we have hydrogen bonds forming in areas like that. And then this would be your alpha helix, it's kind of twisted there. And what we'd find is we would find hydrogen bonds in various places here, kind of creating and holding that amino acid chain into that spiral shape. So the secondary structure again is the alpha helix or the beta sheets, which are held together by hydrogen bonds between different amino acids on different parts of that chain. Now the tertiary structure of an amino acid looks at how the different alpha helices and the different beta sheets cause bending and folding of that protein. So what you get is you got a beta sheet here, but what you might end up with as a result of this beta sheet here, you might end up with a connection, a bond forming, and I'll talk about this bond in a minute, a bond forming between this amino acid here and this part of the beta sheet. Same with this here, or perhaps some type of bond here, or some bond here, or bond here, what have you. So this is how the entire amino acid sequence then bends and folds and twists in response to the order of the amino acids and in response to the beta sheets and the alpha helices that are part of that protein chain. Now a lot of proteins just stop at this third level of structure and then they are free to then do what they need to do. But some proteins enter into what we call a quaternary structure, and this happens when multiple protein chains, multiple proteins in their tertiary structure join together to form this very, very large molecule, which we would call this a macromolecule. In fact, a protein is a macromolecule. If we go back and look, this is amino acids are a molecule, one of those organizations of living things, one of those levels. And then we take those molecules and we join them together to form a macromolecule. Well, we can make a massive macromolecule here by taking multiple protein chains in their tertiary structure, joining them together to form one massive protein with multiple protein chains joined together. This example that you're seeing here is actually hemoglobin. That is an example of a protein in its quaternary structure. And hemoglobin, you should know, and we'll talk about it later in the semester, is what allows oxygen to be, the majority of the oxygen to be carried in the blood to the cells. Also plays a role in carbon dioxide transport and actually playing a role in buffering the blood. Before I forget, these little bonds that are formed in various places during this tertiary structures are called disulfide bridges. And these are formed by sulfur molecule or sulfur atoms that are kind of forming bonds on different parts of the chain. So disulfide bridges. All right, I want to address enzymes here because enzymes are really important. And enzymes are a type of protein or a function. One of the things that proteins can be are enzymes. And enzymes facilitate chemical reactions. They actually allow bonds to be formed or bonds to be broken. And specifically what they do is they reduce the amount of energy required for chemical reactions. They make reactions occur much more quickly. If you were to take glucose and fructose and put them into a test tube and let them sit for a very long time, eventually you would get this molecule sucrose. But by adding them to an enzyme, that reaction occurs much more quickly, and we can produce that sucrose from the glucose and fructose much more quickly. Now, enzymes are kind of neat in the way they work, I think, because they work kind of like a lock and key mechanism. Enzymes are very specific for the reactions that they're part of. So we have the enzyme, and then we have the molecules that the enzyme works with. Those are called the substrates. And the shape of the protein, the shape of the enzyme, determines what those substrates are going to be. Again, example of form and function. And the location in the enzyme where these substrates fit is called the active site. So we have the enzyme, we have the substrate, we have the active site. What we can see is that the substrates will join and fit into the active site here, and then they will undergo the reaction, and then here is our new molecule here. So here's our original substrates, here's our new product here. Enzymes are unique because they do not get consumed in the reaction. In other words, the enzyme does not become part of the final product. When you bake a cake, does the cake pan become final part of your product? No. You're just using that to facilitate the chemical reactions that are occurring between the ingredients in your cake. And that's the same thing with enzymes. So enzymes do not get consumed in the reaction. In fact, enzymes can continue to be reused over and over to facilitate more of the same reactions. So you don't need a lot of enzymes to facilitate a reaction between the same substrates. The enzyme can be used over and over and over again. Of course, the more enzymes you have, the more of these same reactions you can take that can take place, but the enzyme doesn't get consumed, and so you don't need a lot of them.